Welcome to the deep dive.
Yeah.
Today, we are cutting through the noise on something that affects all of us. I mean, truly, every single day. The safety of the food on your plate. Look, it's a simple fact of modern agriculture. To feed billions of people, we need to protect our crops. That means pesticides. They fend off insects, weeds, pathogens. But there's always a trade-off, right? These chemicals can leave behind tiny, tiny traces in the food that makes it to our tables. So today, we're going to unpack that crucial balancing act.
And the stakes really couldn't be higher. You know, we need those pesticides for crop yield, but when the residue levels go above the legal limits, what we call MRLs or maximum residue limits, they're not just traces anymore.
They become a hazard.
They become a potential toxicological hazard. We're not talking about a little bit of dust here. We're talking about compounds like organophosphates or certain neonicotinoids, which have been linked to some serious concerns, neurotoxicity, endocrine disruption. It's the cumulative risk, you know, the little bits of exposure that can build up over a lifetime. That's what keeps regulators up at night. So our mission today is to pull back the curtain on the whole system in North America. The rules, the agencies, and the incredible technology, mass spectrometry, that's used to find and control these invisible risks.
Okay, so we have this massive industrialized food chain. If there's even a possibility of tiny amounts of these chemicals getting through, control is everything.
Yeah.
Who's actually setting the rules here in North America? Who are the authorities, and how did the U.S. and Canada handle it?
Well, we rely on two incredibly detailed sort of parallel systems. In the United States, the main gatekeeper is the EPA, the Environmental Protection Agency.
Okay.
Their job is to define those MRLs, the maximum limits. And this isn't guesswork. They run exhaustive toxicological evaluations, exposure models, all to figure out the safest possible limit for every chemical on every single crop.
So they write the rule book.
They write the rule book, but then actually running the checks on the ground, that falls to the U.S. Department of Agriculture, the USDA. They have this vital program called the Pesticide Data Program, or PDP.
PDP, got it.
Think of the PDP as a constant nationwide survey. They're the ones actually measuring what residues are present in the food chain, from the farm all the way to the grocery store. It gives us a real-time snapshot of what's going on.
That system sounds pretty comprehensive. But I've heard criticisms in the past that maybe the PDP focuses a lot on raw foods, raw commodities, and maybe misses things in heavily processed foods or imports. Is that a fair point?
That is a very fair point, and it's exactly why the system is always evolving. The PDP gives us that robust data on produce, yes, but then you have agencies like the FDA stepping in with their own surveillance programs that specifically look at processed and imported foods.
So it's layered.
It has to be. The system is always adapting to cover those more complex products and, fill in those supply chain gaps.
Okay, so switching gears a little bit, how does Canada's system compare to that?
Canada uses a very similar structure, a check and balance system. You have the Pest Management Regulatory Agency, the PMRA. They're the ones setting the tolerance levels based on their own risk assessments.
So they're like Canada's EPA in this context.
Exactly. And then the Canadian Food Inspection Agency, the CFIA, acts as the enforcement arm. They're out there doing the testing and surveillance to make sure everyone's compliant. But what's really fascinating here is the international connection. Both the CFIA and the PMRA work to align their standards with global ones, especially the Codex standards.
And that's about trade, I assume.
It's absolutely fundamental for trade. I mean, if you're a big food manufacturer, you need testing that satisfies the U.S., Canada, and global rules all at the same time. That common analytical rigor is what builds the bridge for cross-border trade.
So we know the rules are complex, the limits are incredibly strict. And that brings us to the real analytical challenge. We're looking for hundreds of different chemicals at levels so low, we're talking parts per billion, that it's like finding one specific grain of sand in an Olympic swimming pool.
Yeah, that's a good analogy.
So why can't we just use standard chemical testing? Why is mass spectrometry the gold standard now? This is where the tech gets really interesting.
It all comes down to two things, really. Volume and precision. Your standard chemical tests are just too slow, and they simply don't have the sensitivity to find all the different types of pesticides at those incredibly low levels. They might tell you something is there, but not what it is or exactly how much.
Okay.
Mass spectrometry, or MS, basically acts like a highly sophisticated chemical fingerprinting system. We first use chromatography, that's the C in the acronyms, to separate all the complex stuff in the food sample.
Like sorting the ingredients in a soup.
Exactly. Then the mass spectrometer, the MS, weighs those individual compounds with just incredible accuracy, and it breaks them into unique fragments to confirm exactly what they are based on a huge database of known chemical fingerprints.
All right, let's break down the two big techniques. First, LC-MSMS. The LC is liquid chromatography.
That's right. LC-MSMS, so liquid chromatography tandem mass spectrometry. This is the absolute workhorse for any compounds that are water-soluble or for pesticides that would just break down if you tried to heat them up.
Right.
So we separate them in a liquid phase. It's indispensable for things like neonicotinoids or glyphosate. And the tandem MS part, the MSMS, that means the machine does a second level of confirmation.
A second check.
A second check. It weighs the molecule, breaks it into pieces, and then weighs the pieces. That two-step verification is what gives us that part per billion precision and pretty much eliminates any false positives. It's why the FDA can trust the data.
Okay, so LC handles the water-soluble stuff. But a lot of older pesticides, they're not. They're hydrophobic. They stick to fats and oils. I guess that's where the other big technique comes in. GC, MSMS, gas chromatography.
Precisely. If you're dealing with volatile compounds like the older organochlorines or pyrethroids, which tend to be more oily, you just can't use a liquid phase effectively.
It wouldn't work.
It just wouldn't work. So for those, GC-MSMS uses gas chromatography. The sample is heated up, turned into a vapor, and separated in a gas phase before it hits the detector. This gives us the powerful separation we need and the same level of absolute confirmation that the LC system gives us for the other pesticides.
So you really need both systems.
You need both. Integrating LC and GC workflows is the only way to get comprehensive coverage across the entire chemical universe of pesticides. It's a huge investment for a lab, for sure, but it's really the price of consumer confidence.
That precision is incredible, but it leads to a big question. If we're testing down to these tiny levels for hundreds of compounds, who's paying for all that? I mean, is this making safe food too expensive? And what's happening with the technology to make this faster or, more affordable?
That's a critical challenge, absolutely. The cost is high, but the cost of not testing, of non-compliance, recalls, brand damage, public health crises, that cost is astronomically higher.
Good.
So manufacturers see this as a necessary investment. And luckily, the technology is getting faster. We're seeing huge gains from what we call multi-residue methodologies, or MRMs.
Mm-hmm.
Instead of running a bunch of different tests, you can now have single-run assays that detect hundreds of pesticides at once. That saves time, materials, labor. It helps manage some of that cost pressure.
And what about precision? Is that improving too?
Oh, absolutely. We're seeing breakthroughs with high-resolution mass spectrometry, HRMS. Think of systems like Orbitrap. These machines weigh molecules with such insane precision you can identify them down to the subatomic level. We sometimes call it a forensic chemist's time machine.
A time machine, why?
Because the data is so rich, you can do retrospective analysis. Let's say you ran a test 6 months ago and you weren't looking for compound X. With HRMS data, you can go back to that old file and search for it, and the data is precise enough to identify it correctly. It's a game changer for finding new or even unregistered compounds.
And I'm guessing robotics are helping on the lab side, speeding up the prep work?
Oh, massively. Automated sample prep systems are essential now. These robots handle all the messy, complex steps, the extraction, the cleanup that used to be done by hand and were prone to human error. Automation just boosts your throughput and your reproducibility.
So even with all this amazing technology, labs still face some huge real-world hurdles. What are the big technical problems that complicate this kind of testing day to day?
The single biggest analytical headache is something called matrix interference.
Matrix interference?
Yeah. Basically, the food itself is the problem. The natural components in the food fats, sugars, the deep pigments you'd find in something like curry powder or botanical extract, they can actually trick the machine.
Also,
They can either suppress the signal of the pesticide you're looking for, making the residue look lower than it really is,
Right.
or they can enhance the signal, causing a false high reading. This means labs have to develop these incredibly complex cleanup protocols to get rid of all that noise before the sample can even be analyzed accurately. You have to clean out the junk before you can find the target.
and then you've got the administrative nightmare of regulatory divergence. If the science is getting so good, Why are the rules still so different between countries?
That is a major pain point for manufacturers. I mean, the US EPA and Canada's PMRA are trying to align, but real differences still exist, especially when you factor in global standards. Imagine you're importing a spice blend from South America.
What?
It might meet the Canadian limit and the international codex limit for a certain pesticide. But if the US EPA's limit for that same compound is 10 times stricter, suddenly that product is illegal in the US. And that one difference creates a whole separate complex compliance strategy. It just adds time, cost, everything.
Okay, so we've established the incredible precision that comes from LC-MSMS and GC-MSMS. What does all this data actually mean for you, the consumer? How does a lab report translate into a safer supply chain?
It translates through robust risk management. At a high level, this instrumentation is the backbone for the big government programs like the USDA's PDP and the CFIA's monitoring. That's essential for national oversight, but it goes way beyond that.
For individual companies, you mean?
Yes, for companies, testing becomes a proactive tool. It's not always about running a massive screen for 500 pesticides. Sometimes it's about a high specificity test for just 1 or 2 problem compounds in a really tricky product.
Like what?
Like targeted testing for glyphosate in oats or for ethylene oxide in sesame seeds or imported herbal teas. Those require highly customized methods just to find that one needle in that one specific haystack.
So it's less about just getting a pass-fail check mark and more about designing a whole strategy to avoid failure in the first place.
Exactly. The best labs partner with their clients to develop these risk-based monitoring strategies. It's a critical distinction. It involves looking at supplier risk, which regions or which suppliers have a history of issues, and then you align your testing frequency to verify that you meet the specific limits for the EPA or the PMRA. It's an active, forward-looking process, not just a historical measurement.
Right.
It gives that manufacturer the confidence to say, yes, this shipment meets the rules for the market I'm sending it to.
Can you give me a concrete example? How has this kind of high-res data actually forced a company to change its strategy?
Sure, think about something complex like botanical extracts or essential oils. Historically, a company might have only tested those for, say, 50 or 60 known pesticides. But when they start using HRMS on their samples, they might suddenly detect 3 or 4 unregistered fungicides that were used overseas but aren't even tracked by North American regulators.
Whoa.
Now, their own data is flagging a chemical hazard they didn't even know existed. That forces them to immediately drop that supplier, redesign their entire sourcing protocol, and maybe even start talking to regulators about setting a standard for this new compound. The data goes from being a passive check to active, preventative supply chain security.
What we've really detailed today is a complete scientific revolution in how we define food safety. The power of mass spectrometry combining both LC-MSMS and GC-MSMS gives us a level of precision and confidence that was just unthinkable a generation ago. It's a critical line of defense for all of us.
And that line of defense is getting smarter and faster all the time. The future of this field is all about continuous evolution. Beyond just high-resolution machines, we're seeing the integration of machine learning, AI, to help identify peaks and detect unknown compounds we might have missed before. It's dramatically boosting our forensic capabilities.
And what else?
And what's really exciting is the push towards making the testing more sustainable and portable, field-deployable MS. Imagine being able to screen for contaminants in near real time, right at the harvest or at the import dock, instead of waiting days for a lab result.
That brings us to our final thought for you to chew on. If high-resolution mass spectrometry and machine learning are getting so good that they can detect emerging or unknown compounds, things that regulators haven't even made a rule for yet, how might that completely change the regulatory approach itself? Could we move away from relying only on these defined static lists of MRLs and move towards a dynamic real-time risk assessment where the science itself defines the hazard as it appears? That is the ultimate question for the modern food chain.